Hello, everyone. Welcome to today's live broadcast, Recent Advances and Applications of Gas Chromatography Vacuum Ultraviolet Spectroscopy. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're very pleased to bring you this educational web seminar presented by LCGC and sponsored by VUV Analytics. VUV Analytics is the world leader in vacuum ultraviolet, or VUV, absorption spectroscopy. Compounds in the VUV spectrum can be unambiguously identified and quantitated in a variety of industries without the need for chromatographic baseline resolution or continuous calibration. Unlike legacy GC detectors, VUV spectroscopy allows for a more automated analysis with lower risk of errors, shorter chromatographic runtimes, and higher analytical throughput. For more information, please visit vuvanalytics.com. And the University of Texas at Arlington is a growing research powerhouse committed to life-enhancing discovery, innovative instruction, and caring community engagement. An educational leader in the heart of the thriving North Texas region, UT Arlington nurtures minds within an environment that values excellence, ingenuity, and diversity. Whether providing an unparalleled education for its over 56,000 students or addressing important societal challenges, the university is in the business of improving the quality of life in Texas and beyond. Of UT Arlington's 200,000 alumni, about 65% live in North Texas and contribute to the university's annual economic impact of $12.8 billion in the region and $13.6 billion in the state. For more information, please visit uta.edu. Before we get started with the webcast, we have a few housekeeping announcements. First, the webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, which you can find on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the webcast. If you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. And at the end of today's event, we will invite you to complete a brief survey provided by our sponsor. The survey will automatically appear on your screen, and we hope you will participate. Before we get started, before I introduce today's speaker, I'd like to ask you all to participate in a brief polling question. You can click directly on your screen to submit your answer. And the question's up there now. What analytical challenge provides the most difficulty in your gas chromatography methods? Isomer differentiation, analyte coelution, peak identification, compound quantitation, isobaric analytes, or non-ionizing compounds? Once again, you can click directly on your screen to answer. What analytical challenge provides the most difficulty in your GC methods? Isomer differentiation, analyte coelution, peak identification, compound quantitation, isobaric analytes, or non-ionizing compounds. Okay, let's pull up those results. Oh, we've got answers kind of all over that variety of answers there. I guess compound quantitation is number one, and analyte coelution is number one. Compound quantitation on its heels there. Excellent. Thank you so much for completing that poll. I would now like to introduce today's speaker. We're very pleased to be joined today by Dr. Kevin Shug. Since joining UTA in 2005, Kevin Shug's research has been focused on the theory and application of separation science and mass spectrometry for solving a variety of analytical and physical chemistry problems. Two main research threads are currently pursued in their group with some intertwined aspects. One is fundamental studies of chromatographic separations and mass spectrometry ionization mechanisms, and the other is the isolation, characterization, and trace analysis of bioactive compounds in complex matrices. Kevin Shug's group is also active in chemical education research. He has over 100 peer-reviewed publications and over 400 presentations, posters, and invited talks to his group's credit. Kevin Shug is also the director of Collaborative Laboratories for Environmental Analysis and Remediation, or CLEAR. And he has received awards for his research and teaching, and he is a member of several editorial advisory boards. Kevin Shug also writes a regular blog for LCGC on various aspects of separation science. Kevin, thank you for joining us today. You have the floor.
Thanks very much, Laura. I appreciate you moderating this and appreciate LCGC hosting this. I'm really excited to be here to talk about what's been going on in the GC-VUV world. I think I gave kind of a first webinar on this a couple of years ago, and it's pretty astounding what we've been able to, what we have and others have been able to really accomplish in this short period of time. So I'm going to go ahead and get on with it and talk about some of the research that we've been doing in close collaboration with VUV Analytics and other partners. I think the easiest starting point here is just to recognize that the use of absorption detection for GC is really a minor part of the use of GC detectors compared to what people are used to in terms of LC, and I think it's quite obvious why. It's that gases don't really absorb in the UV region. You have to get down below the kind of traditional 200 nanometer cutoff that we're used to considering in LC to start seeing the absorption of most gaseous species. So, if you think back about why that is, you have, of course, these different types of bonding in molecules and different functional units, which are going to give rise to electronic excitations when you absorb light. And so in the UV vis range, which is highlighted there, so from let's say 200 up to 700, we can see that a lot of these highly functional compounds, things that are aromatic and whatnot, absorb quite well, and that's quite a large amount of analytes. But when you start to get into gas phase species and things that are very volatile, many of those things can lack that functionality. So alkanes, for example, have no absorption. That's because they're primarily dominated by sigma to sigma SAR transitions. And so we really have to get down into this vacuum ultraviolet range. And so I'm highlighting there some 120 to 230 nanometers, 240 nanometers, where this instrument that I'm going to talk about today is operating. So, like I said, this not only gives us rise this ability to look at compounds that we couldn't see previously with a traditional UV source, but also gives rise to the ability to look at more intense transition for other compounds. For example, benzene, we're used to looking at 254 nanometers. Well, its absorption at about 180 nanometers is actually about 10,000 times higher than it is at 254 nanometers. So there's some really significant advantages to being able to look at the absorption of compounds in this range. The instrument we're talking about here predominantly is the VGA 100, and this is just a schematic to kind of walk you around through this. Basically all chemical species absorb in this wavelength range and have unique absorption spectra, but luckily carrier gases are largely VUV transparent. They absorb, but they absorb very little. And so they make for a good background for carrying the GC separations in through a transfer line. You have basically a deuterium lamp source that's supplying light through reflective optics through this flow cell. The flow cell is thermostated at 320 degrees, and then you have absorption that occurs that is essentially based on the Beer's law principles that we're all familiar with from traditional UV vis spectroscopy. So quantitative analysis is straightforward, and then we have these very rich featured spectra. You're going to see a lot of those in the next 30, 40 minutes here to be able to do some unique qualitative analysis. The technique is non-destructive, so it could be placed in line with a mass spec. I'm going to talk a little bit about that at the end with a new version of this detector. And then we're collecting all of these spectra simultaneously at a rate up to 100 hertz. So you can do very rapid chromatography. I'll show a little bit GC by GC work. And then post-run, we can do a lot of filtering and processing of that data to pull out the qualitative and quantitative features of interest. So just to give, if you haven't ever looked at any VUV spectra, this is I think a really interesting point here. I just want to point out that I'm showing a lot of these spectra as normalized absorbance on the y-axis. So this is not depicting the relative absorption of different compounds that we're comparing. In many cases, compounds of similar class have similar absorptivity, but in this case, for example, I mentioned nitrogen has a very low background. It has a very neat and rich absorption spectrum, but it's quite low cross-section or absorptivity overall. But you can see really some of the rich features that are in some of these compounds on the left, and on the right, as we take methane and start substituting out chlorine atoms and increasing the chlorine atoms, you can see how the richness and the variability of that spectra changes quite a lot. So what's been done so far? As I mentioned, it's been a very busy past couple of years, and I'm mentioning stuff that we've done here, and as well as what I understand to be kind of what's out there from other researchers currently. I'm hoping I haven't left anybody out, but certainly looking at gasoline and different fuels, the ability to look at permanent gases, pesticides, fatty acids. We're really trying to look at really the top applications in the GC and demonstrating where the VUV can be very complementary to traditional, let's say, mass spec analysis. There's been some work to do some kind of fancy data analysis, automated deconvolution. That's this time interval deconvolution. I'll talk about that a bit more later for gasoline analysis and looking at some polychlorinated biphenyls. We've done some fundamental studies looking at limits of deconvolution. I'll also talk about that. And then others have focused heavily on characterization of things like diesel fuels and breath volatiles. We've also looked at quantitative aspects and collaborated with others in particularly in this designer drugs and work. Collaborated with researchers at George Washington University and in the Czech Republic at Palacký University to study how the VUV could be used there. And then at VUV themselves, they've done some work looking at how they could quantify water and solvents. So I'm going to try to give you a little bit of snapshot of this. Cited on the bottom there is a fairly recent review that covers a lot of this work. So you can go there to refresh over or look at some of the things maybe that I don't have time to cover. In those works, we kind of highlight these key features, unique but class-similar spectra, excellent complementarity to mass spec, so we can see things that mass spec has problems with, like isomers and different cis-trans arrangements and Labile compounds. As I mentioned, it's pretty much a universal detector, so requiring this full range of absorption, but then we can apply spectral filters on the back end to do some selective detection of different classes. We can deconvolve co-eluting analytes, which is probably one of the most impressive and useful characteristics of the detector, and we can do this in an automated fashion. I'll talk about that. I say respectable quantitative performance. I mean, low to mid picogram on column is nothing to sneeze at. Sure, ECD may be more sensitive or some mass specs. It's about as sensitive as a mass spec currently in a scan mode, which is plenty good for many applications, as I'll show you. And then I'll also talk a little bit about how we're using theoretical computations to support this. So just to kind of go through quickly a little bit here, some examples, and we've shown these before, but places where mass spec has some problems. Here's showing some spectra, VUV spectra and mass spectra of some isomers of, let's say, the xylenes or naphthol, and I just want to point out that on the left there, those VUV spectra are extremely distinct. We have resolution down to about 0.5 nanometer, and so any cases where you see any kind of crossover in these spectra really shows them to be differentiable. So in the gas phase, these are very different. On the mass spec side, these are very similar, and if we have something like a coelution of meta and paraxylene, we don't have to worry about separating them chromatographically. We can just simply deconvolve the contributing signals to from each of those different individual components to that overall signal. So it's very powerful to be able to take some of the burden off of the chromatographic separation. We looked early on at some pesticides and some interesting ones here. Here's a series of the hexachlorocyclohexane isomers, different pesticides, and again, you can imagine quite complicated mass spectra, quite similar mass spectra, due to the high isotope content on these things, but very unique VUV spectra. Captan, Folpet, and Captan are kind of thought about as kind of the big three challenges in terms of GCMS pesticide analysis. These are N-trihalomethylthio antifungals, and these are very difficult to detect by GCMS because they essentially just fall apart in the electron ionization detector. But we were able to show mid-level picogram detection limits and a very nice rich absorption spectra for monitoring these compounds by GC-VUV. And then, low mass species permanent gas analysis. We had a really interesting opportunity to work together with our Department of Electrical Engineering here to look at the thermal runaway of different lithium-ion and lithium metal batteries. Obviously, aircraft really don't want to have that happen in the cargo hold. So we kind of looking at what the things were produced by different types of batteries, and many of these things are difficult to see by GCMS, but the VUV is able to look and see, at oxygen, carbon monoxide, carbon dioxide, ethylene, all the way up to some other more toxic compounds, acetaldehyde. It certainly became apparent that you didn't want to have these things undergo thermal runaway, but here was a nice way for us to simulate that and use the detector to kind of characterize which different chemistries or battery chemistries could be more susceptible to releasing, let's say, toxic or flammable gases. And then a lot of attention has been paid to FAMEs, fatty acid methyl ester fatty acids. That's because we have the ability to really differentiate cis and trans isomers. So two examples here of different isomers that are very similar mass spectra but very different VUV spectra. The FAMEs themselves are very interesting. I mean, you have, if you start kind of with a saturated FAME there on the left, it has kind of more like an alkane feature. And yes, we do probably need to separate those chromatographically to differentiate them if we get to the very high carbon number. But as you start adding in saturation and cis-trans isomers, you get a lot of these featured spectra up around this 165 to 205 nanometers, and it makes them quite distinct and easy to be analyzed. And so I think a lot of interest and work on this topic of FAMEs and various matrices for GC-VUV. I was able to, I had the, it was really a tough situation. I had to go and spend several months in Sicily on a sabbatical with the Mondello group, and I know you feel bad for me, but this shows a little bit of example of what we got done there. I was going there to learn some multidimensional separations in GC-GC and LC-LC, and we were able to. They were pioneering some work of some flow modulated GC by GC, which has problems coupling to mass spec because you're using such high flow rates to develop the second dimension run. And so in this case, flow rates up to 12 mils per minute were no problem for the VUV because it's a mass-sensitive detector. So essentially, flow rate independent sensitivity. You're not diluting out the signal by going to these high flows. And so we focused on some complex mixtures, some fatty acids. That group is really, kind of world-renowned for work in food analysis and certainly lipid analysis. And so shown on the kind of the top right are some traces of various different lipids, fatty acid methyl esters, and you can see how the patterning of the separation by the nonpolar column in the first dimension and the polar column in the second dimension really give you a nice, let's say, practical picture of the different saturation and unsaturation that are present in this particular sample. Down on the bottom left, you can see we apply this to like a biodiesel, and so along the bottom lines, you have all of these saturated alkanes going up the chromatogram, the 2D chromatogram. You see all these different monoaromatics, diaromatics, and triaromatics, and you can see those fatty acid methyl esters really popping out there. And so you have this, very nice resolving power of the 2D GC, and then you couple on that another dimension of the VUV absorption, and it becomes a very powerful combination. So I think that this is something that's going to develop more, and I was really happy to be able to work with them in that capacity to show some of the first work on that. So you get into the fuels analysis, and this has really been an interesting topic and one where the VUV is really latched on. This is just showing some examples of compounds that are commonly present in fuels, so linear alkanes or paraffins, cyclic alkanes or naphthenes, branched alkanes and olefins and aromatics. And you can see that, okay, the paraffins are this fairly similar absorption spectra, but as you start to add in some branching or cyclization, you start to see some wiggles and additional features in the spectra. When you really start to see major features is when you start to add some unsaturations to the olefins and the aromatics. And so using these traces, then we could start to, the analysis of gasoline was really a very first start for us, and we've expanded on that significantly. This is just showing, by the way, an application of the spectral filters. So we've collected the full scan absorption of this gasoline sample. And then if we apply a spectral filter from 125 to 160, we can accentuate the absorbance of the alkane or the saturated compounds, versus if we want to apply this 170 to 200 nanometer filter, we can really accentuate the aromatic absorption component of the sample, because those are absorbing preferentially in that range. So this gasoline proficiency standards became an interesting part to go forward and develop further. People mentioned earlier on in the poll that co-eluting species are a problem. Well, here's one example from the gasoline where we had a co-elution of 1,3,5-trimethylbenzene and 2,3-dimethyl-octane. So in the middle and the bottom there, you see a square around that co-elution. In the orange is shown the full range absorption. And then we have a couple of spectral filters where if we apply the blue low wavelength range, we accentuate the absorption of the saturated compound, or if we apply in green the 170 to 200 nanometers, we accentuate the absorbance of the aromatic. And then because we have this sum spectra over that place, which is shown up there in the inset B, that mixed spectra can be easily deconvolved to the contribution from each of those components, and then we can, as shown in A, essentially project the individual components post-run just by simply deconvolving the contributions. It's a very simple additive spectra that can be where you can back out the individual components, assuming you have those components in the library. Well, where can we take this a little bit further? Well, PIONA analysis of gasoline, so characterizing finished gasoline is a big deal. It's a big business. There's a lot of gasoline produced around the world. But there are a whole bunch of ASTM methods to do this type of characterization. Well, because we can capture all of this data and have all this class-specific organization, we've shown that you can do all of this PIONA analysis, so essentially potentially replacing 10, 11 different ASTM methods using one single analysis that can be done in about 35 minutes, and that's also a savings in time. So this is using a concept called time interval deconvolution, which I want to tell you a little bit about. This is basically a notion of taking the chromatogram and chopping it up into slices, and then considering the absorption of different components at each slice, and essentially bending those out into to collect them into the different classes of interest, to then later process to convert into amounts and whatnot. So this has been applied for finished gasoline, and very recently was approved as an ASTM method. And then we've also done this for some Aroclor mixtures for polychlorinated biphenyls. So just to show you this a little bit quickly, to do this, what we need is a reference library spectra for the analytes of interest. So consider, let's say, the PIONA molecules. We'll take the different types of compounds that are present in gasoline, and we'll segregate them by class. We'll say, okay, that's a paraffin, that's an isoparaffin, that's an olefin. We'll put that information in the library. Some unknowns are allowed because you have very similar absorption spectra for different classes. But obviously, the more that you can cover, the more accurately you can classify the sample. We'll assign a retention index for each compound, and that's basically just so that we can start to use specific retention indices to pare down the library that we're searching in to assign different compounds in the complex mixture. And then to convert those absorptions into, let's say, a mass percent or some amount at the end, we assign a relative response factor for each compound or class of interest. So let's say we've run our sample, let's say our gasoline, and so we'll set some retention index tolerance. And let's say, okay, the retention index, say plus or minus 25 to 40. That's a pretty liberal tolerance. And again, all that we're doing that there is to pare down the library that we're going to search for when we're trying to match the absorbance to the different classes and compounds. We'll set the time interval that we want to slice our chromatogram up into. So let's say we're going to consider every 0.03 minutes. And then we'll go through the slices sequentially. So we'll find the first slice and calculate the total absorbance. We'll select the reference library that corresponds to that reference index window, and then we'll perform some tiered library search for the different components that are giving rise to the absorbance there. If necessary, if there's 1, 2, 3 compounds, they'll be deconvolved. And then that absorption will be binned into the different class or species that we've specified, and then we'll proceed to the next time interval and do this in an iterative fashion until we get all the way through the chromatogram. And then we'll convert all the absorbances to relative response factors. So here's just a quick snapshot of what that might look like for the front-end run of a gasoline. So you see we're kind of going along, no absorption, no absorption. We might say that, okay, something there is, we'll set that as a background to allow us to do some subtraction. But coming up close to there, 1.35 minutes, we get our first absorption. Okay, there we've got 0.074 absorption units of 3-methyl-1-butene. So we'll bend that into, the olefin units, and then we'll move on to the next one. The next one's got a little bit of that 3-methyl-1-butene and a little bit of isopentane. So we've got a little olefin, we've got a little bit of isoparaffin, and we move on and so forth. And so we move down through this sequentially, and just basically, we don't have to consider a particular chromatographic peak. We just have to consider the absorption that's occurring in a particular time slice, and then assign that absorbance to the correct compound. And so this can be done extremely rapidly. After, let's say, a 35-minute run for this gasoline, within 30 seconds we can spit out the PIONA classification. And to cut to the chase, this is showing what that PIONA analysis by the VUV method, which is now an ASTM method, is performing compared to other ASTM methods. So there was a lot of people who contributed to this data, and really appreciate their contributions to running these, and so we could do the comparison. But along the x-axis there are a series of 13 different gasoline proficiency samples, and on the y-axis are different measures that we might be interested in. And this is just a selection. We have other measures like olefin content and whatnot. But the important point here is that the dotted line tracks along all those colored lines, which means that from each proficiency sample to the next, we get excellent agreement between the VUV ASTM method and the other ASTM methods that are there using this time interval deconvolution. So really powerful automated way to treat the data. We looked and extended this to looking at commercial Aroclor mixtures. These are mixtures of polychlorinated biphenyl. So there are 209 different ways that you can put chlorine around these rings, and then there were various different commercial mixtures that were made for different industrial applications. These have now been banned because they have, are carcinogenic, have endocrine disrupting properties, and there's a significant interest in being able to characterize these, particularly in the environment. What we decided we would do is look at these as a compound class, and it turns out that all 209 of these PCBs have unique absorption spectra. And this is just showing a selection of some of them, the actual ones that correspond to the EPA method mix. But we ran all the different Aroclors and applied this time interval deconvolution to see how well we could match the degree of chlorination that would be present in there. So along the top row are different designations of Aroclor mixtures, and that second number indicates what percent of chlorination should be in that sample. So they would run these, they prepare them, they'd catalyze the chlorination of biphenyl for different amounts of time to get different degrees of chlorination. And you can see there on the very bottom, so we decided we'd classify these based on their content of mono, di, tri, tetra, so on, different biphenyls. And then we can add those up, and you can see that our determined total chlorine content is very much spot on to what it is supposed to be according to the commercial mixture. So again, very rapid analysis and deconvolution of these complex mixtures. So this notion of deconvolution is a big one, and when you have two compounds that are very different spectral features, very different spectra, they can be easily deconvolved over a wide dynamic range or wide range of abundance when they're overlapping. But when spectra becomes similar, then there's going to end up being some limits. And so we decided we were going to study those limits. So we picked a set of compounds that have historically been challenging, so dimethylnaphthalene isomers. And we decided what we're going to do is going to make some different mixtures of these in known amounts and see how well we could deconvolve them so we could understand the limits of the range where we can deconvolve them. We needed some way to quantify spectral similarity, and so that's represented by the sum squared residuals in the bottom. So basically, if we compare different dimethylnaphthalene spectra against each other, if we subtract them from each other and square them to get this sum squared residual, that becomes some number that can quantify spectral similarity. So the lower that value in that table, the more similar the spectra. So you can see that we have some range here. The highest one between, let's say, 1,4 and 2,6 is 42, so those are quite dissimilar. But then if you look at 1,2 versus 1,6, the SSR value is 0.6, so those are very similar. So the question is, if these are overlapping, which they are challenging to separate, to what degree, at what point do we lose the ability to deconvolve them? And to make a long story short, this is a very nice paper. This is one paper that we got is on the cover of Analytical Chemical Acta at the end of last year. Really nice fundamental study looking at these mixtures, and we basically could show that down to about a 99-to-1 ratio, and of course you have to consider how much is on column, we could reliably deconvolve the contributions of these. So you can barely see it there in the top left, but as we go to 595 and 10 to 90, you can see the relative contributions of these 1,4 and 2,3 dimethylnaphthalenes as we deconvolve them. So we found it's kind of a rule of thumb when you get down to this spectral similarity that's exhibited by the dimethylnaphthalenes, that you can get about a 2 order of magnitude overlap. As long as you have a significant amount of that on column, of course, that number is going to be go down as you get down closer to the detection limits, but starts to understand how we can use spectral dissimilarity to differentiate these things. We expanded that very recently to look at a whole set of new designer stimulants in collaboration with George Washington University and Palacký University in the Czech Republic. We collected a set of 43 designer drugs, some 11 sets of isomers. So these are compounds that are out on the market that people have produced to try to sidestep analysts. So they basically want to make bioactive stimulant compounds that the analysts are not ready to look for. So you're talking about bath salts and legal high and all kinds of different drugs to try and avoid some legal repercussions by selling something that's legal and not illegal. But the problem you get here in the analysis, and this is exemplified with these two isomers, this pentadrone and 4-methylcathinone, is that if we look at the collision-induced association on an FTICR mass spec, very difficult to differentiate these two isomers. And so the idea was to go into these sets of compounds and see how the VUV would do. So looking, just to cut to the chase, if you look at, here's a set of 4 different isomers, 4 different isomer sets. So you have all the isomers that are 177 point blah, blah, blah. So you can see the elemental formulas. They all have common elemental formulae within these different sets. And whereas the mass spectral differentiation of these would be quite difficult either by CID or electron ionization fragmentation spectra, the VUV spectra are quite distinct. And we went through these and even used the same type of sum squared residuals concept to look at how similar or dissimilar these are. We even did a bit of work looking at deconvolution of these, and of course, even if you had 2 similar things like this, 2,3-dimethylmethcathinone and 4-methylbufadrone, they could be quite well deconvolved. And then, on the left, we're showing the kind of the sum spectra in the blue on the top, the individual contribution from the different components on the bottom, or the bottom 2, and then that dashed line is if you were to sum together those 2 spectra on the bottom, how well it matches the experimental spectrum. So, just kind of perfect pulling out those different individual components.
Kevin, this is Laura. Every now and then you're breaking up a little bit. I don't know if you are on a headset and can switch to a handset, or I'm not sure. It's kind of going back and forth. So I don't know if there's a change on your end or not.
I am on a pretty old phone here, handset, that's been very reliable.
Okay. You sound okay now, so I want you to keep going then if there's something you can change. Thank you.
Okay. Thanks for letting me know. Hopefully it's coming through well, and I apologize if it doesn't. If you miss something and have a question at the end, you can certainly always contact me if there's something, and hopefully you'll have a copy of these slides on the end as well. So one of the very interesting aspects of that about the VUV is the ability to think about using computational methods to support the experimental determinations. Now, previously, most of the VUV work was done in synchrotron facilities, so you can imagine there's a lot out there, but not a ton of research that's been done in collecting experimental spectra. So, I would say that the theoretical computations to follow on to the experimental work is kind of let's say lagged behind. And now that we have access to the ability to really collect experimental spectra for a lot of different compounds very quickly, then it makes sense to see whether we can use these computations to support the experimental work. So just as a little idea of how this works, when we compute an absorption spectra, what we're doing is predicting an excitation energy based on a structure. So that gives the wavelength and the peak position. And that's just a line at a particular point on the wavelength. And then we get an oscillator strength, which is the intensity of that signal. Now those lines are then, as you see on the red there on the bottom, are then fit with Gaussian functions to kind of simulate the broadening that we get through vibrational coupling and whatnot from experimental spectra. So they're going to be much smoother, and it is a bit of a reconstruction there. But you'll see how that works. Typical computation of UV vis is difficult because in solution it's very environment specific, and so it's hard to get a good match with the computational work. In the gas phase, of course, we have better predictive power because it's a much simpler background. So just to kind of go through some of these, we were trying to find the right approach to do this, and time-dependent density functional theory appears to be the best one. So in the red there is the benzene absorption feature, and then you see the feature that's predicted by different computational methods. And so it's always a game of accuracy versus computational costs. And in this case, it's fortunate that these TD-DFT calculations are quite fast. Some of the higher level calculations that take you a lot more time, I mean, who wants to wait 10.5 days to calculate benzene? And it's not so useful for us if we're trying to do a lot of these things. And it's not nearly as accurate in this case. There are some cases where these higher order computations can be more accurate, but in general, this time-dependent density functional theory is fast and pretty accurate. There's a fairly regular tracking with the experimental spectra. So here's to give you an example. I showed these type of antifungal pesticides before, and you get some idea of how the computed spectrum in blue can match the green spectrum, the experimental spectrum, and as I said before, that those red on the bottom that's those oscillator strengths and intensities that we're using to fit the Gaussian functions in blue. We applied this to the dimethylnaphthalines, and you can see there some interesting, very nice, it's really quite good correlation between the experiment and the theory for these very similar spectra. But it's not perfect. There are some shifts. So the dark lines, the experiment for a couple of these overlaid with the theoretical, which are the dotted lines. And so you can see there's some shifts. But it does quite well to reproduce trends. And just to give you one more example, we use this in the new Designer Stimulants. And you can see for a series of these isomers, for the 236 AMU isomers, the top two and the one on the bottom left are comparing the experimental with the theoretical, and then on the bottom right, I'm overlaying all of the theoretical spectra. So what could be very interesting here is that, and where I could really see this being useful, especially in this realm, is that we could onboard the spectra for compounds even before anybody ever made that compound. So if you thought about, what is the next designer stimulant that somebody is going to make, in principle, we could go and compute that spectra. We could have it sitting on a GC-VUV software, and if it shows up, at least, it's not going to match something that's already been there, but if it starts to match some of these additional, maybe other signatures of, let's say, unknown or never been synthesized before compounds, that could give some idea of, hey, maybe this is another newer designer stimulant that we need to be looking at and take a closer look at. So I think the VUV really complements the ability to use mass spec and other techniques, especially in combination in these forensics realms where you'd really like to have as much information as possible. So I'm almost done. I got a few more slides, but just to summarize here, I hope I've shown you that VUV is a really nice complementary tool to mass spec. You get these very unique class-specific signatures, things like isomers, isobars that if you have problems with mass spec, these are easily differentiated. I didn't show much quantitative work in this case, but we do have that in many of the papers, and happy to discuss that further. But the fact that you have a Beer's law correspondence to the absorption means you can that absorption increases very regularly. You get nice linear responses. All of the advantages of quantitation that people are used to in LCUV, we have that for the GC-VUV, and then some. We have the qualitative aspects of the spectra, and we have the fact that all these absorptions are done in the gas phase. We can register cross sections for molecules, so a molar absorptivity that's never going to change into the database, so that every time you saw an absorption event, you know exactly how many molecules are there. That's a whole other story called pseudo-absolute quantitation, but there is a reference to that earlier in the paper, earlier in the talk, if you're interested in that. Obviously, I've shown deconvolution of co-eluting species. I mean, this does provide less of a burden on separation. I know, in terms of GC column manufacturers, there's a lot of work done to create custom columns that can separate particular isomer pairs that mass spec couldn't differentiate. The VUV provides another window to looking at that and reduces the burden on the separation there. So you can get a lot more done with just your standard column. And then, as I showed, we can also, for complex mixtures, use this automated deconvolution, this time interval deconvolution. Lastly, the theoretical computations is something that's coming along and being developed. I think it's going to be a really unique tool to aid the analysis here. Mass spectrometrists do this type of thing when they're doing like an infrared multiphoton dissociation in the gas phase, where they can model these things and using experiment and theory, and it's very powerful. So the combination here of the theory and experiment, I think, will be equally powerful going forward. The last thing I just want to show is what's next. And this instrument now is commercially available. I've talked about the VGA 100. This is the VGA 101, and it has some interesting additional features. The first and foremost, they've extended the spectral range from now the higher end is not 240, it's now 430 nanometers. And so this is really advantageous as you start to look at, let's say, these polyaromatic hydrocarbons or compounds that have high degree of conjugation where the absorption maximum is shifting out above the 240 nanometer range. So you can see here very regularly as you go to higher ring polyaromatics how that major absorption signal now moves out into this range that can be monitored by the 101. Here's a whole series of different isomers. These are very common compounds to be that are desired to be monitored. Some may be pyrroline is considered carcinogenic, and so these are the C20H12 PAH isomers, and you can see how they have very rich absorption features. And back where we could measure, but even as you go now to this higher spectral range, you get that additional spectral feature information, which I think is going to be really important, especially if you start to monitor these things in an environment. Environmental samples where you'd love to be able to make sure you can pick an absorption region that has unique absorption for those compounds of interest, but where your background does not absorb appreciably. And then in order to accommodate being able to analyze some of these bigger compounds, they've increased the temperature stability limit up to 430 degrees. So they're showing, here we're showing some chromatogram where it's tailing off up to something like C96 hydrocarbon because of the ability to look at the high temperatures. And then with some improvements in the light path and the lamp and how that's positioned, we get a higher sensitivity in this system. So getting down below into the single-digit picograms on column. So I think this is going to be a really interesting instrument to work with, and as it gets more out into the market and people are starting to investigate it. But probably one of the most desirable aspects was the ability to hook it right between a GC and a mass spec, so it has the ability to accommodate the two transfer lines so that you can now put these instruments in series and have that complementary information together in one analysis. So looking really nice going forward. I can't wait to get mine to play with here. So with that, I definitely wanted to leave some time for questions, but I certainly need to thank some people, and that's a long list of people to thank that we've worked with, so I'm not going to read them all, but certainly had a lot of interaction with VUV, support from Shimadzu, friends from the Czech Republic who've been having various student exchanges, and I've been going there, and they've been coming here, and it's been a great collaboration. Also collaborators in Messina, Manny Garbalina and Valero helped a lot with the PIONA analysis. Ira Lurie and his students at George Washington with a lot of the designer drugs, and on the top right is a list of a long list of names of people whose work I get to take credit for here giving this talk. And certainly like to add my thanks to Paul Johnson from VUV for helping organize this webinar, and also to Laura for moderating. So with that, I'll hand it back to Laura and to wrap this up, answer some questions.
Thanks so much, Kevin, for your excellent talk. Before we go to Q&A from the audience, we have one more polling question for all of you who are out there and watching. And once again, you can click directly on your screen to answer. So that question is up there now, and the question is, what are your typical chromatography run times? 5 to 10 minutes, 10 to 15 minutes, 15 to 20 minutes, or longer than 20 minutes? So once again, you can click directly on your screen to answer. What are your typical chromatography run times? 5 to 10 minutes, 10 to 15, 15 to 20, or longer than 20? All right, and we'll pull up those results.
Wow.
Ooh, so the vast majority are you doing with run times longer than 20 minutes. Very interesting. All right, well, let's go right into our questions here. As a quick reminder, if you want to ask a question, you can type it in the Q&A box, which you can find on the right-hand side of your window. Okay, let me pull up the first question. So, Kevin, has there been an attempt to quantify experimental spectra using theoretical calculations? And if so, how have the results turned out?
So attempts, yes, I actually say that's a very interesting aspect that we're working on. How can you theoretically predict the cross sections of the intensity of these oscillator strengths? How well do they represent the experimental spectra? I don't think it's been done yet, but we're looking at it to see how accurate it could be. So it's a very interesting idea again, but to my knowledge, that's not something that's been shown yet definitively.
Okay, thanks. If you have a triple coelution and the third component has a spectrum intermediate to the other two compounds, can you still deconvolute?
Well, okay. The first answer to that question is, yeah, 3, 4 components, no problem to deconvolve them. Again, given the caveat that they have, it depends on the relative abundance in which they're present and how similar the spectra are. Now, if you have a spectra intermediate to another one, what initially jumps to mind is some situation where you'd have some interconversion between two things kind of colluting on the column. So, that would give rise to these what are called, Batman peaks, or having, ability to see interconversion on the chromatographic timescale. I could imagine there that it would be really interesting to see, as you go from, let's say, a product to a reactant that's happening on the column, you're going to have some transition state in the middle that's going to have some intermediate spectra, and so that might be hard to differentiate at first. But certainly, if you have, let's say 2 of those components, and you have their unique spectra, the third one, even if it's intermediate, is still unique. And so, if you're able to log that particular spectra in the library, then certainly you'd be able to deconvolve the different contributions.
Excellent, thank you. If you were trying to quantitate by compound class, for example, trying to answer the question, how much isoparaffin is in this sample, do you have to have compound-specific standards?
So Not really. There's a very, and again, this is covered quite extensively in the paper on the PIONA analysis that I talked about. But yes, different classes of compounds can have different absorption strength. Now, one of the nice things is about the absorption is that across different compound classes, you really don't have a huge variation in absorption. So you can pretty well predict how well something's going to absorb looking at structure. If you consider an isoparaffin class or any other class, when you start looking at within class and let's say a homologous series or a building off of that series, the absorption changes the strength of absorption changes in a very regular, reproducible fashion. So indeed, it would be quite possible to, without having, as long as you could appropriately classify a signal as an isoparaffin, and when you get out to really long chain things, maybe that starts to get more complicated. Actually, I know the naphthalene is where, when you get to bigger carbons, essentially assigning that spectra for, let's say, something above C13 as a naphthalene might be more challenging. But if you were able to say, I've got, these are signals for isoparaffins, without having a standard, certainly you do a very good job of saying, I've got this much isoparaffin in there, without needing the standards.
Excellent. Have you run any nitrated aromatics or aromatic oximes? And if so, what are their behavior?
Yeah, I would have to say that that is not a class that we have looked at closely in my lab, so I couldn't answer that one specifically. But whoever asked that question, feel free to send me an email, and I'll find that information for you.
Great, thank you. What is the highest number of co-eluting compounds that you've been able to analyze using the VUV spectroscopy?
I'm quite, again, that goes kind of to the whole notion of spectral similarity. We tried to go a little bit crazy with those dimethylnaphthalines and create some great degree of coelution. And if everything was present in the same amount, we could easily deconvolve 5, 6, 7 of those from each other if they were overlapping. So that's talking about a class that's very spectrally similar. Now, if you started to vary the relative abundance of those, I think you'd start to lose the ability to distinguish them. But in the PIONA classification, for example, I believe the code is set up to go up to 3 or 4 different coelutions, and that seems to be plenty in that case. And it performs very well. So I'd be happy to say the number, 4 to 5, but it actually has not been practically tested to the extent of being able to answer that question directly and saying it's exactly this much, because it very much depends on how spectrally similar those overlapping compounds are. The more dissimilar they are, the more things that you could deconvolve.
Great, thank you. This next question, I'm not entirely sure about the wording of the second part, so I'll read it as is, and maybe you can help me figure it out. So why does it have to be VUV and not just UV for GC? Is it because the molecules in gas phase that VUV is applicable to GC?
Well, so I guess there's a couple different ways to answer that question, and so I'll just start talking at it and see. Hopefully I hit the right answer. So the notion of UV is really, I guess it's just us as chemists have, or I guess I don't know who did it, the physicists, but have kind of defined this range of, let's say, 180 to 400 nanometers being the UV range, and then below that, historically, let's say below 200 or 180 or whatever, has been called the vacuum ultraviolet range. And the name, the moniker vacuum, and we don't have a vacuum on the system, we're just using a low absorbing background, but previously in say a synchrotron facility where you wanted to make measurements in this vacuum UV range, you would have to evacuate the environment so that you did not have background absorption from air. Now we can make absorption measurements in air in the UV range if you've got something that can absorb there, because the air does not absorb. So it's really a semantics there in terms of the VUV and UV. But of course, in this instrument, in the 100, we're going out into the UV range, so it's technically a VUV spectrophotometer. And as you get into the newer version 1, you're going even further out there. I mean, some might argue you're even starting to get into the visible range of the analysis. So I think it's more semantics why we call it this. The fact of the matter is that they've come up with a very nice way to be able to look at, VUV absorption on a benchtop system that was not previously possible, and so that's opening up a whole different range of possibilities relative to what was possible with GC before.
Great. Let's see if we can fit in a couple more questions here. Is the VUV wavelength stable enough for multivariate data analysis from multiple runs of similar samples, such as similar chemicals with different composition?
Definitely. So we've started to look at some of those, and I actually do get that question quite often. I don't think it's been explored nearly as much as it could be, and I guess that's probably part of the fact because the absorption, the class-specific absorption, allows you to not need necessarily any very sophisticated statistics to pull apart the different classes. I mean, that information is inherent to the different absorption spectra for different classes. But to answer the question directly, yes, the signal is extremely stable. Whereas, let's say in a traditional UV on your LC or benchtop UV with a cuvette, you can see the absorption maximum shifting a little bit from run to run, depending on how much water was in your solvent or whatnot. In this case, in the VUV, in this instrument in the gas phase, those absorption positions of, let's say, absorption maxima or absorption features are really rock solid. And I alluded to that before, where I said if you can bank a cross section, which is essentially the molar absorptivity across that whole absorption range, you can put that in the library, and it will be constant whether I run it today or I run it, next month or next year. That's a physical property of that molecule, and it will not change. You may go up or down. It will definitely go up or down a little bit depending on concentrations there or how much you get into the system. But the ratio and the ratio of different absorption features and their position is extremely reproducible and constant. So you could, I think it will be a next step when you get into more complex mixtures to apply more of these multivariate methods, but it has not been done yet, but it could be.
All right, let's do one more. Can the technique be extended to gas analyses of both hydrocarbon and chemical gases?
Yes. So, and I don't mention it here because we haven't really played with it much, but the company does offer what they call an SVGA, but it's essentially an online gas analyzer. And I don't know the specifics of where these would place it, but imagine having a production gas or something that's flowing through this thing and continuously monitoring, and you're worried about a certain impurity popping up. So it's absolutely could be used to monitor for presence of trace impurities in a bulk stream, or just quality control of a bulk stream, and let's say a continuous process. So certainly is capable for use there, and they have a product to do it.
Fantastic. Thanks so much, Kevin, for your excellent talk and for answering all those questions. We have to wrap up now, but thank you.
Oh, and my pleasure. I hope everybody enjoyed that.
Also want to thank all of you in the audience for attending and participating in today's event. And I'd like to thank our sponsor, VUV Analytics, for making today's webcast possible. As a reminder, at the end of today's event, we'll invite you to complete a brief survey provided by our sponsor. The survey will automatically appear on your screen, and we hope you'll participate. Today's program will be available for on-demand viewing through April of next year. You'll receive an email from LCGC alerting you when the webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.